The overall goal of this microscopy-based in vitro assay is to study actin polymerization from endosomes in vitro.
This method can help answer key questions in the membrane trafficking and actin fields, such as the role of actin in the endocytic pathway. The main advantage of this technique is to reconstitute the nucleation polymerization of F-actin on early endosomal membranes in the test tube, thus rendering this complex series of reactions amenable to biochemical manipulations. Demonstrating the procedure will be Jorge Larios and Olivia Muriel from the lab.
Begin by placing dishes of HeLa cells onto a wet metal plate in a flat ice bucket and washing the cells twice with 5 milliliters of ice-cold PBS. After removing the PBS from the last wash, add 3 milliliters of ice-cold PBS per dish. Then use a flexible rubber policeman to scrape the cells off the dishes in a quick, circular motion around the outside of the dish, followed by a downward motion in the middle of the dish. Scrape gently to obtain sheets of attached cells. Next, use a plastic Pasteur pipette with a wide opening to gently transfer the cells to 15 milliliter conical polypropylene tubes on ice. Centrifuge for 5 minutes at 175 times G and 4 degrees Celsius. Gently remove the supernatant and add 1 to 3 milliliters of homogenization.